Thank you for attending this seminar, and we were very lucky to have Olga Amelkina here. She offered herself to do this seminar here, and it's a great opportunity because we are not paying her anything. But she was around Seville because she's collaborating with the Iberian Lynx project, and she's an expert in reproductive physiology that became trained in reproductive physiology, but now is getting into genomics. So now she is a good candidate to trying to merge, understanding reproductive physiology with omics tools, and that's the subject of your talk today. Just to mention, she's coming from the IZW Institute in Leipzig or Berlin. This is Institute for Zoo and Wildlife Research. The institute and the groups in there have been collaborating with the Iberian Lynx project since the start of the captive breeding, supporting all the reproductive aspects of the Iberian Lynx breeding. So yeah, and now Olga is joining the group after staying at the Smithsonian lately, where she also collaborated with the black-footed ferret breeding program, I guess, and conservation program in general. So thank you, Olga, and the floor is yours.
Thank you very much for the introduction. Thank you everyone for coming. The title of my talk is System Biology of Fertility, because I want to show you how to approach the study of fertility from different sides. So, because systems biology is the way to study the field from not just focusing on one isolated part, but looking at it as a network. And now that we have all this generation of different layers of the omics data, right, from genomics to transcriptomics to proteomics, metabolomics, lipidomics, we have this chance to really see the more complicated picture of it and use this systems biology approach, which has been used a lot in a lot of human research and the medical research, but I would like to bring it more to the animal, also reproduction research and the wildlife research. And the reason looking at fertility, of course, it's very close to us, to me and Laura, who are also from my Department of Reproduction Biology, because we're looking at the reproductive biology, adaptation of the animals, but also reproductive management of the animals. But I'm pretty sure it's also close to everyone working here, because it relates to evolution, it relates to population health, it's all relates to the fitness of the animals and individuals. And today I want to kind of tell you 4 short stories, kind of examples of this combinational approach of system biology to study fertility. And I will start with the fertility preservation studies in felids. This is the work that I have been doing together with the Smithsonian Institute at the Center for Species Survival. And there will be a lot about reproduction, which I hope would be, I will introduce you to the reproduction world, which hope to find interest in you to connect it back to the genomics. And then I will talk a bit about the black-footed ferret research, and there were 2 big projects. One was focusing on the environmental effect on the black-footed ferret ejaculates and their reproductive success. Another one was looking at the genetic load, and we were trying to predict the fertility trends in the black-footed ferret. And finally, I'll say a couple words about something new that we're starting in the Iberian lynx projects from the trying to study the reproductive adaptations of the Iberian lynx. So when we talk about fertility preservation, right, it can go on different levels. One is to preserve the habitat, right? So this way, of course, we're preserving the fertility of the intact population. Another one is breeding, captive breeding centers, where we try to facilitate the natural breeding in the animals. This way we also preserve the fertility of them and genetic diversity. But when this doesn't work, we have to go to the artificial reproductive techniques. So for example, there have been a successful artificial reproductive techniques applied in the clouded leopard with the artificial insemination, and in the cheetah with the embryo transfer technique. It was just in 2020 was the first successful embryo transfer technique in cheetah. And the reason why sometimes we have to go to those reproductive management techniques is because of the species specificity. For instance, in the clouded leopard, if you breed the 2, male and the female, if they live together since they are little and they know each other, they breed normally in captivity. But if you're trying to introduce the male and female that don't know each other, for example, you want to combine the 2 genetic diversities from them, then because the clouded leopard is so stressed, it's such a stress reactive species, they usually fight they are too aggressive to breed. And so in this case, you have to collect the semen from the male and artificially inseminate the female. And in the cheetah, sometimes on the female side, things may not be, for example, the female may not go through a full cycle or full ovulation, and this way you have to do the in vitro fertilization and then do the embryo transfer. And all of this is supported by the preservation of fertility by freezing, right? We all know about freezing the gametes, freezing the sperm, freezing the oocytes, or the embryos, but also freezing the tissue, for example, freezing the testicular tissue and ovular tissue. And the benefit of the freezing of those parts of tissue, even though it's more complex because tissue contains many various cells and you have to preserve all the connections and communications between them, but testicular tissue has spermatogonia, so just the beginning of the gamete production cycle. And the ovarian cortex contains the dormant follicular pool. And this means that you can sample individuals from young to old, out of season, in season. So you always have this pool of genetics that you can preserve and then later on develop tools to mature, for example, the spermatogonia or the follicles, and then use them for artificial reproductive techniques. But to mature them, you have to figure out how to do it, and so that's why we have this amazing model for the wild felids, which is the domestic cat, because we can have access to the domestic cat gonads from veterinary clinics. So whenever they spay or castrate the cats, we can get testes or ovaries as a byproduct of the ovariectomies or gonadectomy, orchidectomies. And we use this model a lot to test different preservation methods. Usually the preservation is using liquid nitrogen, right? We all know about the cryopreservation. There is types of slow freezing, or there are types of fast freezing, which is vitrification. But they do have their limitations because, of course, it's a high cost. You have to have access to liquid nitrogen. It's prone to human error if you don't have this system that refills liquid nitrogen by itself. It's also not field friendly. Not every place can have access to liquid nitrogen. So at the Smithsonian, we have been actually looking at different ways to preserve the cells. And we've been having inspiration from nature, which is the phenomenon known as anhydrobiosis. Basically, it means life without water. So there are many animals that can lose over 99% of water and still remain alive. One of the biggest example, of course, as we all know, is the tardigrade, which can dry out and then come back to life. But it's not only that, it's the dower larva of the C. elegans, some species of rotifers, the embryos of brine shrimp, some of the anthropods, and even one of the teleost, which is the annual killifish. The embryos of this fish, they live in these ephemeral ponds. So when they dry out, the embryos go into kind of sleeping stage, they dry. But then when the rain comes back, the ponds are filled out, the water is there, and they're alive again, the reproduction starts again. What unites all of the species is that they all were found to use trehalose, which is a disaccharide, it's a sugar, and it seems to be a really good protective of the cellular components. So once they start losing the water, the trehalose vitrifies, so it kind of crystallizes and envelops all of the cellular structures, stopping all the metabolism and keeping membranes intact. And so we got inspired by this, and we were thinking, okay, why don't we make domestic cat cells into little tardigrades and see if we can use that then to develop the new preservation technique. And this was, the beginning was led by Pei-Chi Lee at the Smithsonian Institute in the Pierre Commissolis group, where in the beginning they found microwave-assisted desiccation technique where you load the tissue, or if you're working with sperm or with the oocytes, the cells, you load them with trehalose and then you dry them in the microwave. It's a special microwave that makes sure that it's uniform drying of the cell. And then you can keep it potentially at room temperature of 4 degrees, and then you rehydrate it and the cells are back alive. And so when I joined, I started developing this technique more in the tissue, so in this ovarian tissue, to preserve the primordial follicles and also the spermatogonia in the testicular tissue of the cat. But you cannot really do it as trial and error, right? You can change protocol here, change maybe cell permeabilization, but you're kind of doing it blindly. And so we decided to look at it in a systematic approach. And so we did some of the transcriptomic studies to see what is happening. And in the Pierre Commissolis lab, what we did, we exposed the tissues of the domestic cat to various techniques of preservation, either to drying for different amount of time or to the cryopreservation. And then we measured the response, the transcriptomic response of the tissues to see how they react to the stress of the preservation. We did it in the ovarian cortex and the testicular tissue. And then we went to another level. So this in RNA sequencing, we looked at the overall gene expression. And then we did the microRNA sequencing to see the regulation of this expression. So if we do see the difference expression, are microRNAs involved in it? Because microRNAs are the small non-coding RNAs which are known to inhibit the gene expression either by degrading the RNA itself or inhibiting the translation. We did it also in the ovary and testicular tissue, and then finally we combined together the networks, doing the microRNA, mRNA interaction networks, trying to see what is actually happening in the tissue in response to it. And this is the first network that I'm going to show you, and I will spend a little bit longer on this, but just know that all the next networks will kind of follow a similar idea of it. So you will always see the legend of the network, and the legend will tell you what the node is. In this case, it's the circles, and what is the edge. In this case, it's the connective, so connections between them. So this is the results of the functional enrichment of the differentially expressed genes between two groups. In this case, it's vitrified, so cryopreserved group, and the fresh tissue. And the red here color means upregulated, so these genes were upregulated. Blue is downregulated, these genes were downregulated. The functional enrichments, it's kind of looking at what the overrepresentation of the terms. So we have upregulation of these genes, of the set of these genes. Are they overrepresentative of some pathways or functions? And here it's really summarizing it. So it's you have the circles, the node, which is the functional term, which is enriched functional term. You have the size, which shows you the bigger the size, the more genes are involved in this pathway from our data set, the more genes are enriched. And then you also have the color on the border. In this case is how significantly it's enriched. So more yellow, kind of lighter color is more significance. And finally, in the edge, you have the overlap size, because all of these nodes are connected by sharing the genes. For example, here you have some of the mitochondrion or other respiratory chain, and they're all sharing similar genes, the same genes, and they are all enriched together, which comes into this cluster of mitochondrial respiration. So we could see that, after vitrification, actually our cryopreservation induces this increase in mitochondrial respiration, which later on, when looking at the literature, is apparently an adaptive response of the tissue to combat the stress. We then looked at the protein-protein interaction network. So this is a different type of network. In this case, the node is the gene itself. And then, so here's differentially expressed gene, and then the edge between it is the interaction score. So this comes from the string database that hosts predicted and validated protein-protein interactions, and you can then build it in the cytoscape with your own overlay of your data. So here, for example, the red is fold change, so upregulated genes, and blue downregulated, and the border is also significance of the differential expression. And we could see here, you see this cluster of the genes that are tightly interacting with each other. They're all mitochondrial actually related genes. They're all from the mitochondrial genome also, and they're all enriched in the mitochondrial respiration. Now, looking at a little of a bigger network, this is already the effect of drying, and you can see a lot of blue here because a lot of signaling is going down. So drying had a more detrimental effect because this protocol is not optimized yet, and so we could see lowering of the functions in cell adhesion, in the signaling, and also specifically in this RAS signaling, which we saw in the protein-protein interaction network as well. So all of these genes, their proteins are not predicted to interact with each other, and they're all enriched in the kinase signaling pathways. The next step, we overlay the microRNAs, right? So we put in our microRNA sequencing, and here we're looking at the upregulated microRNA. So here in the middle, you see, it's difficult to read, but it's MIR182 and MIR183, the two microRNAs with 5P arms. They're upregulated, and all of these little kind of balls around them, these are downregulated mRNA targets, because microRNA connects to the mRNA to inhibit it, and so predicting what these microRNAs are targeting from our data set, from the downregulated mRNAs, I could build this network where we really found that specifically these two upregulated microRNAs seem to be controlling the response in the tissue to drying. What is more interesting, you can then overlay the next layer of data. This is overlaying the predicted protein interaction to it. So in the green, you have the predicted microRNA mRNA interactions, but then overlaying it in purple, you see protein-protein. So it's microRNA mRNA kind of protein-protein interactions, and you see that all of these regulated, down-regulated targets, they're also predicted to interact with each other. So it gives us more confidence that most likely these two microRNAs are really controlling the response of the tissue. And then the next step, what we could do, because all of these targets, what are they enriched in? What are those functions? And so we can extract all of these targets and then do again the functional enrichment, and then this time visualize it by linking to each of our microRNA. So as you can see, for example, this cluster of the ATP binding and protein kinase domains, they're all regulated by these two, 1A3 and 1A2 microRNAs, and they're enriched in this. But then you have the transcriptional regulation of polymerase II, and you have additional microRNA also involved in regulation of this. And this way we kind of can decode what these microRNAs may be regulated in our data set. So that was the part of fertility preservation. Now I'm moving on to another story. It's about the black-footed ferret, and I think it's a species that we know. It's one of the success stories together with the Iberian lynx, where it was termed extinct twice in the last century, but through the in 1980s, through the work of a lot of conservation biologists and scientists together, it was possible to bring the last ferrets they found some of the ferrets in the wild and brought them in captivity. 7 individuals became founders, unfortunately all related in a way. They all have the kinship of 0.12 or something, so they're kind of first cousins, similar to that. And that's why, even though they've been released to success story that they have brought back to the wild, but they still need management from the human side. So because their reproduction is not great, their semen quality is not great because they went through this bottleneck, and they seem to be in captivity specifically. They seem to be experiencing the environmental dependent and breeding depression, because when looking at the comparison with the wild population, so this is for the captivity. So for example, in the wild, you can see this is in 2017, they're checking the normal sperm, and wild born had a much higher normal sperm than the captive born individual. So we notice that the wild are more, they have a higher fertility. And in captivity, the whelping success, which means that a percentage of female that bred and also gave birth, has been declining. And one of the hypotheses, which was done by the Rachel Santemeyer, who is the one who's involved in the reproduction breeding of the black-footed ferret, one of the hypotheses that she found is that we see this decline in whelping success, decline in normal sperm production, and around the time of the decline, there was a change in diet. So the captive ferrets, they switched from these mink pellets, where they also added the rabbit meal, to the standardized commercial diet for small carnivores. And to compare, in the wild, over 90% of black-footed ferret diet is prairie dog. So similar to Iberian lynx is the rabbit, in the black-footed ferret is the prairie dog. And there was a decline. And so there was the hypothesis what is happening there. And one of the hypotheses that because the control diet, the store diet, is really high in vitamin A, perhaps it has this effect. The higher vitamin A can limit the absorption of vitamin E. It may lead to the oxidative stress in the sperm. So perhaps this can be the reason for lower quality. And so the experiment was done, which was done by Rachel Santemeyer and her PhD student at the time, Nadia Ali, and together with Klaus Köpfli and many Vasquez, and then inviting me also to join this interesting project. We looked at wild, so we had several groups of the domestic ferret, or black-footed ferret, which is the wild population, who just use the prairie dogs and some of the small rodents. Then there was a population captivity of carcass only, which tried to emulate the white diet. Then there was the control, which just eats normal, the tore diet. And the vitamin E supplementation, because the hypothesis was that perhaps vitamin E can combat the stress, the oxidative stress that we observe from the diet in the sperm. And then there was some mixed also diets. And when we looked at the sperm quality in the beginning, when we looked at the motility, so this, the motility of the sperm was similar, but then the normal sperm again was higher in the wild, and the acrosome, abnormal acrosome, so it's another seminal quality parameter, you could see that the abnormality was higher in the control groups. And what we did the proteomics and transcriptomics of the ejaculate of this black-footed ferret, so collected from captive and wild. And what was very interesting in the beginning, so this is looking at the proteome of the ejaculate of this ferret, we saw that, all right, there were 739 proteins shared between all of the groups. But what was interesting, that the wild group had 149 proteins found only in them. And that's to remind that this is only 4 animals, and the rest are 22 animals. All these 149 proteins were not found in their ejaculate. We looked at the composition and previously reported that it was combination of the sperm and seminal plasma proteins. And now, and then we looked at the overall proteomics of all of the proteins, and this is just example, kind of a small deviation just to show you that when you try to do the predicted protein-protein interactions with thousands of genes of thousands of proteins, you get this hairball. It's pretty discouraging, and you don't know what to do with it. But there is a way to handle it. You just need to use some of the different clustering techniques. So in this case, I use the Markov cluster algorithm to kind of detangle it based on their interaction score, so how more closely they interact with each other. And once you do that, you can start seeing clusters. Each of them are then enriched in different functions, for example, mitochondrion or structure or motility of the sperm. And you can look around, and so we kind of created this atlas for the ejaculate and the black-footed ferret of the proteins. We see which ones are expressed, which ones were previously reported in other species. Now returning to those specific proteins of the wild. So here, only 110 proteins of those were in the database of the protein-protein interactions. And this is using the domestic ferret database. So this is kind of translating using domestic ferret background and the database on the black-footed ferret. And so when we looked, so these are the proteins found in the wild control carcass vitamin E diets, and wild only were this 110 proteins. And then if we tried to look at the enrichment of those clusters, we found that they're enriched in ribosome and translation. And that was interesting because, well, it's not really directly related right to the sperm quality, but we know that the wild individuals are more successful in breeding. And so looking through the literature everywhere, what is happening there, we found a hypothesis is that apparently the sperm, it's not just bringing the genetic material, it also brings translational machinery to the female tract, so it helps for the embryo when it's formed to jumpstart translation and to develop more rapidly. So perhaps the wild individuals actually have an additional ribosomal machinery. So others, of course, they still had the ribosomal proteins, but in this case that perhaps the success of the wild individuals in the breeding is not at the term of the sperm motility, it's happening at the point of embryo development, which we cannot really measure. We don't see it, we're a little bit blind to this, but we see what the sperm is bringing in, and this can be one of the contents that we can compare. So then when we looked at the transcriptome, so sperm is transcriptionally silent, so there is no transcription happening apart from sometimes in the tail and the mitochondrial genome. So this, that when you look at the transcriptome, you kind of see what the sperm either bringing also to the female tract or what happened to the sperm before when it was maturing, and maybe there's still some RNA left. And so we looked at the transcriptome and we compared between the diets. There was not that much association between the diets, but the biggest association we found when we were looking at the factors of the diet is when we put in the factor of the successful or failed breeding. So we have all of the information and data for all of the ferrets, if they bred, was it successful or not, and then we could, each samples, we would put, was it successful, was it failed, was it unknown, and then adding this factor actually gave the biggest association for our transcriptome expression. And it was interesting because as you see, so in red, these are all enriched in successful samples. This is blue, is the failed samples, and we again see some of the things related to translation and ribosome, and this is when we do the protein-protein interaction network, we again see the translation initiation cluster being enriched there as well. So it's kind of supporting a little bit the hypothesis we saw in the protein, but we need more, of course, studies to overlay those things. And so the next project on the black-footed ferret is going a little bit a different direction. So this one we really looked at the ejaculate of the black-footed ferret and really at the functional studies. And here we go back to more of the genomic studies, and this was the project of Enrique together with the Klaus Koepfli, where they sampled 54 individuals from different populations and then analyzed, so they sequenced them and analyzed the genetic load in them with different methods. So this is the VCF fire clustering in the PCA plot, and you can see, so there are 6 wild derived, but out of those 6, so these 4 were one of the related founders. So they were related to the founders, some of them were, to these captive and reintroduced populations. And then these 2 guys, SB2 and SB10, these were the ferrets that were not successful at breeding. So they did breed, but they didn't, I think one of them bred and the progeny didn't survive further, so didn't bred further. So this line kind of got cut off. And this is part of what the Black-footed Ferret project is trying to do, is they are trying to clone these individuals and bring back these genetics. And I think everyone heard the news that there was some successful already cloning from this SB10. So one of these samples where there was 3 clones that survived, Elizabeth Ann, Noreen, and Antonia also could breed further. So that was interesting. So we'll see, maybe these genetics can be brought back to the black-footed ferret population. What Enrique did first, so we wanted to look at the genetic load, right? So at the deleterious mutation, and there is the SNP effect tool that is known for calculating, and this is the first step. So Enrique he took all of these 54 individuals and looked at the prediction of deleterious mutations. So at the high, separating it by high impact, so some of them are lost mutations, for example, stop codon gained, or moderate, which a lot of missense mutations were characterized as moderate, or low impact. And then I took the data, so I have data from Enrique where I know, okay, these variants were found in these genes, were found in these individuals in heterozygous or homozygous states, and then I kind of took all this information and tried to figure out which functions could have been potentially affected and which of them may be related to reproduction. So at first I looked around, okay, let's see overall at the low, medium, and high. And if you put all of this together, this is also the domestic ferret background, so looking at the enrichment, there are big kind of clusters of functions being enriched. But then you can zoom in and you can see that so this one, the orange one, this is specifically the high impact mutations enrichment. And if we look closer at this, we can see that overall there is some motor activity. So there is accumulation of mutations in the genes related to the motor activity to the cytoskeleton. But then specifically accumulation of the high impact mutations in this DNA chain and microtubule. And it was interesting because sperm for motility, for example, uses the DNA chain. This is part of the cytoskeleton of the sperm. And we found also enrichment in this DNA chain microtubule when we were looking at our previous experiment in the sperm in the ejaculates of the black-footed ferret. And there were some associations between the wild phenotype and compared to the captive one, that they were enriched more in this DNA chain and the microtubule, which may be related to the motility of sperm. Next, looking at this data, what I could also do is just, looking at the different populations, so wild, captive, reintroduced, to see are all individuals from all populations the accumulated mutations which are enriched in this function or not. For example, again, this DNA complex, it seems that majority of wild ones are getting accumulated, this variation, but maybe it's those SB that were separate. So this is something to look deeper in that, because maybe they are already carrying some of the deleterious mutations there. And then there would be some, for example, this MHC class 1 antigen processing enrichment, where there were a lot of reintroduced animals that were carrying variants enriched in it. But this is still in progress. So this is a preliminary data that I'm showing because it's kind of was the first look, because the next step is to overlay more results of the prediction tools. And this is the work that's done together with 2 groups, with Hierarchy Group and Diaz and Fraser Lab, where both groups are using different ways to predict, in this case, specifically missense mutations that may be deleterious. And so Becky Shaw in the group, she identified the genes that had elevated non-synonymous to synonymous ratios, and then annotated them with SIFT to see if these mutations predict to be deleterious, is it predicted to be tolerant, or if it's unknown effect. And 66 out of those variants were predicted to be deleterious. I took those variants and tried to compare it together with what was identified in the SNP effect. And the next one, and this is the results that's also going to be incorporated later on, is the Diaz and Fraser lab, where Paulina, the student, she took using the deep learning models, trying to identify predict the 1% of pathogenicity of the mutations. And I asked, let's look at the 1%, 5%, 10% to just see, again, to build this kind of similar network as I was showing you with the low, medium, and high impact, and to see is there something that we're missing in a more overview, and we can then narrow down on just this 1%. This network hasn't been put together yet. It's still in progress, and hopefully we'll finish the paper soon and be some exciting results altogether. But so far, this is just, again, the preliminary look where I was trying to put the protein-protein interaction networks together from the SNP effect, only the high impact, and then overlay these SIFT results. And here you can see that you can map all the different data that you have. For example, the nodes are, again, these are the genes, right, that were accumulating these variants. The border, so the shape, is whether it's fully heterozygous in all of the population, the three populations we're looking at. Is it fully homozygous in all of these animals that were identified, or is it mixed? Then the border color, you see the animal number. So the lighter one, it means it was identified in less animals. Then inside you can see whether it was identified in all of the population, or like this, for example, RPS10 was only in captive population, identified in captive population. Then we have some identifiers whether it was also identified in SIFT, or is it a known infertility marker. Here we use the mouse and human infertility database. And finally, the interaction score again, and here it's the overlay of predicted functional but also physical interaction. So in red, for example, this is a very tight cluster because it's predicted to interact functionally and physically between the proteins. And this is just excerpts of some of the clusters. The network was much bigger, there were much more clusters, but this ones I just want to show examples of some of the functions that may be related to the sperm function and to the male fertility. For example, we have our DNA motor complex, right, and inside of it there is this interesting gene, topaz1, which was shown to have complete loss of function, predicted to have complete loss of function, and also it's a known infertility marker. And it's homozygous in almost all of the 53 animals. But then at the same time we have this SPATA5, which is also was identified by the SIFT tool, and then we found that it was homozygous in captive but still heterozygous in the wild population. And this was very interesting because Klaus has a project on the southern black rhino as well, where they are also looking at the genetic load, and I got the kind of the list as well of those genes and tried to look around, see what some of the enrichments, and I noticed that there were 14 genes with a high impact shared between the Blackfoot ferret population and this southern black rhino populations, and one of them is this NLP14, which is also a known infertility marker for sperm morphology. So there are some things that we get kind of snippets. This way, in the network, we can investigate, where should we look? What comes next is overlaying the other results, so from the other lab with the machine learning. And the exciting next step is also, we do have the sperm parameters and breeding success for a lot of these ferrets, not for the wild, but for a lot of captive and some of the reintroduced before that, that they were captured in the wild and then measured. For the sperm parameters, and so the next step is to overlay it, because maybe we find this variation in this individual, and then we can see, is the motility actually affected in this individual? Is the motility lower compared to the other ferrets from the captive population? And now that we have kind of experience with it, the next step to look at around in the other species is one of the projects on the jaguar reproduction project, which is done together with Laura, who's sitting here, where we want to see, there's a lot of genomic studies on the jaguar reproduction done by Eduardo Eiseric in Brazil, and they accumulated a lot of very interesting and good genomics data. And what we want to do is, together with another scientist from IZW, Daniel Foster, to sequence the European captive population of the jaguars, which has been in captivity for over 20 years, actually even longer, because they got from US, some of the captive jaguars that came from US. And they're speaking with the coordinator of the species, we know that the fertility has been going down. So the breeding success of this jaguar is going down. So it would be interesting to compare this captive population with the wild population in Brazil, and also with the ex situ population in Brazil as well, because those are kind of closer to, would be closer to the wild population, and compare the reproductive parameters, and also see if we can see any predictions from the genetic load that may affect the fertility. But this is something that we're still starting and trying to also applying for the funding to get and other things, but hopefully you'll get some interesting, exciting results. And finally, I just want to say a couple of words about the Iberian lynx, finally, and kind of coming back more into very, very reproduction specific field, because the Iberian lynx is a really, really interesting species, not because it's a beautiful bearded cat, but also because they have a very unique reproduction adaptation, and it has to do with the corpus luteum. And the corpus luteum, it's a reproductive gland, and it's a temporary gland. It's a gland that appears in the body and then disappears. So it's maybe a very nice target for evolution to play around with, and which I will explain now. So corpus luteum usually forms it forms in the ovary after ovulation. So we have the follicular development. When the follicle is mature, the oocyte is mature, it ovulates, the oocyte goes out, it goes to the oviduct to meet with the sperm for the fertilization. But what remains after the ovulation are the cells, follicular cells, which undergo lutealization. They become luteal cells and they form the corpus luteum. I'll also call it CL sometimes interchangeably. So corpus luteum or CL. And then the animals need, and mammals need, this corpus luteum to produce progesterone to support the pregnancy. In some animals, for example in cats, corpus luteum is the major source of progesterone, and that's how it was discovered actually in rabbits. If you remove the ovaries during the pregnancy, the rabbits were aborting because they needed corpus luteum to support the pregnancy. But in humans, for example, we switch from corpus luteum to placenta secretion. So in this case, you're not fully relying only on corpus luteum. The placenta also supplies progesterone after a certain time. After it goes, it supports pregnancy, and then it regresses. So if there was no successful fertilization, it will go away. If the pregnancy finishes, it will also go away. It's done its job, it's out. And the progesterone level goes down, and this opens a possibility for the new cycle to start. You can start again. What happens in the lynx is different, and this is something that was discovered by our department of at the IZW by Katerina Yevgenov, and together with Frank Goeritz. I'm not sure, maybe 20 years ago over that, when the breeding program was started. They found out that in the Iberian lynx, you have this corpus luteum which starts regressing but never really goes away, and it stays in the ovary, and then there is an ovulation that forms a new corpus luteum, and this new corpus luteum together with the old one keeps staying in the ovary. And what they found that it's not only persists, so it persists for at least 2 years as measured in Eurasian lynx, and it keeps producing progesterone, which may interfere with the new cycle. And it seems that at some point, so you have this kind of annual production of the progesterone, but as the time comes for breeding season, you need the progesterone to lower down to allow for the new cycle to start. And so this is the mechanism we're still not sure. Is it something that's needed? How is happening in the lynx? So it's something we're trying to investigate. How is this corpus luteum is silenced? How can it be regressed? There are some other adaptations you can see in the mammals of this corpus luteum. For example, based on literature, we can find that corpus luteum also persists in hyenas and cetaceans, so some of the whales and dolphins. We are not sure if they're functional or not, and also in hyenas, they have a very specific reproduction with the high androgen levels in females, so maybe it's not similar to what we observe in lynx. There is an interesting phenomenon of accessory CLs that we observe in elephants, because elephants have a 2-year-long pregnancy, so they really need a lot of progesterone. But they only have one oocyte that ovulates. So when this one oocyte ovulates, they also have a lot of other follicles nearby that don't ovulate, but they all become corpus luteum, and then there is this whole group of corpus luteum giving enough progesterone to support this really long pregnancy. Horses actually do that too. They also have the accessory CLs. There are some hypotheses we have why the lynx have this adaptation. One of the hypotheses which we are now actually challenging after visiting breeding centers and talking more with people, and this was the previous hypothesis, was that we know that Iberian lynx, Eurasian lynx, and Canada lynx are all monoestrous, means they ovulate only once a year. bobcat is polyestrous. Bobcat also has persistent CLs, but it's polyestrous, somehow can ovulate several times. But before we measured, we don't really know how high progesterone is in bobcat throughout the year. We just know it persists there structurally. But we know that in Eurasian Iberian lynx, progesterone is still being secreted. So the hypothesis was that maybe this was the adaptation of the lynx to secure monoestricity. That once it ovulates, if it's not successful, if the fertilization is not successful, then that's it. They have to wait for another year. Because if they ovulate later on, then maybe the birth of the cubs gonna happen at the worst time of the year, and they want to avoid that, so they have this monoestricity. However, now we travel around the breeding centers, and apparently, at least in captivity, some of the lynxes started to ovulate second and third time as well. So they're not strictly monoestrous anymore, or they never were. So this is kind of hypothesis we're challenging. Another hypothesis is related to elephants, to the accessory CLs, because there are some observations that the Iberian lynx, their first pregnancy is not always successful. So perhaps they need to first have this corpus luteum from the previous cycle, and only the next cycle, so the second cycle, they already have this kind of persistent corpus luteum, that their own accessory CLs that the whales and horses have, and then now they can finally have a full successful pregnancy because they have all the support of progesterone. So this is something that we're looking at. And before we were looking at the study of the corpus luteum, we were really lucky that we could get some of the ovaries from the two Iberian lynx, which is Azahar and Saliega. They were ovariectomized for health reasons because Azahar is a really great breeder, and she already contributed to the breeding of the Iberian lynx, and it was the last time she got mated, and then she was spayed. And then Saliega, it was a younger female, but she had some health problems, so she had to be spayed as well. And Azahar, actually, some of the embryos of the Azahar, 7-day-old embryos, were cryopreserved also for the future. And this was done by Katarina Evgenov, and from there we could get the two ovaries that together had freshly formed corpus luteum, so just formed 7 days old, and persistent from the previous year. And we were really working on those kind of comparison of this fresh and persistent, adding also some of the CLs we could get from Eurasian lynx, and also characterizing the cycle of the domestic cat to understand how CL forms, maintains, and regresses, and see what is different in the lynx. But at that time, which was over 10 years ago, and this is when I did my PhD actually at IZW, then I left, and now 10 years later I'm returning back to the institute and trying to have this network systems biology approach to it. But at that time before, we really were looking at the very specific system. We could only pick, oh, we're looking at these genes, we're looking here, we're looking at these proteins. We looked at the apoptosis system. Apoptosis system was working well. So it's not that the tissue cannot regress at all. We're looking at the steroid receptors. Steroid receptors were there, they're expressed, so it can react to the hormones. We're looking at steroidogenic capacity. Yes, this corpus luteum have all of the steroidogenic capacity to produce progesterone and estrogen. And prostaglandin system, because prostaglandins are known, for example, in the cow, they have this very active regression signal for the corpus luteum, which comes from prostaglandin 2 alpha. And so we were wondering, maybe this corpus luteum in lynx doesn't have a functioning system to detect this signal, but it does. So we kind of found a little bit of a wall. We characterize it, we know there's something happening, but we don't see the full picture. And so that's why the next steps, this would be the final slide of this presentation, just to show you what's going to come next, and hopefully next time I can give you more results from here. But we want to really do now this systems approach for this. So we still have some RNA and tissue from Azahar and Saliega. We are seeing if maybe we can collect some potentially more samples from ovariectomy, so we can have a higher number of this. We generated domestic cat corpus luteum transcriptome. It was generated by Beate Brown from our lab. So we could really track formation, maintenance, regression in the cat species. We have access to some of the public data sets that generated for other species for corpus luteum. And then we have sometimes additional feline species that come to our pathology, which died in the zoos, and so we can have some of the CLs from there. Now what we're doing is, from all these tissue samples, we're generating different levels of data. We're generating RNA sequencing, and for example, from Azahar, Saliega, already data generated, and also from Eurasian lynx, and just need to analyze it. Then we want to start looking at the microRNA sequencing, because microRNAs are also very tissue specific, and sometimes you can even track the evolutionary trends through gain and loss of the microRNAs. For instance, one of the interesting study that we're doing with another group is looking at the evolution of the microRNAs in placental animals, and you can see sometimes association with the placental types and which microRNAs, specific microRNAs, this group of species has. For example, cow has a cotyloid placenta, which is a very weird kind of island placenta, and it has this microRNA that is not seen in other placental animals. It's only also seen in one of the bat species, which is a bit weird, but we don't know with that association. No one looked at the placenta of that bat species. So yeah, we want to see the microRNAs of the cells because maybe they have the unique microRNAs in the cell there. We're looking at the proteomics, so the kind of final stage of the gene expression. And finally, the next step to look at really at the single cell RNA sequencing to see the population of cells, how different of them are expressing, is it different between formation, regression, and persistence, and putting it all together with the networks approaches that I showed you, overlaying with the domestic cat, luteal formation, regression, and other species, and hopefully decoding this mystery of the CL in the lynx, which can also contribute to the breeding program. I forgot to mention it, that it does have, so it is an evolutionary curiosity, but it also has an application part, because if right now Iberian lynx is breeding well in the captive breeding centers, but some of the individuals do require assisted reproductive techniques. For example, there are some males that maybe due to behavior cannot be bred, and this is where Laura comes in, who is doing the artificial insemination, for example, from those males. Or there are some, for example, there is some frozen sperm, as I know, from the one of the male that never bred, which could also be brought back into the population. But the thing is that you have the female and the female cycle. We need to figure out how to manage the female cycle, because we need to, this is something that's also Laura is actually doing and working on her project, is how do we stimulate the female to ovulate in time so we can do the insemination in time. And if you have this persistent corpus luteum, sometimes it's in a way. So we need to really figure out how to manage the ovarian cycle of the lynx, and decoding corpus luteum will help us. If we have this kind of interference from persistent cells, maybe there is a way to inhibit it and really to make the animal ovulate and be part of the breeding pool as well. So thank you very much. This is, I would like to acknowledge of a lot of people for the first two projects, especially for the fertility preservation and for the Blackfoot ferret diet project, and for the next two projects, hopefully in some years I can give you the update and some news, and especially for the, I think, Blackfoot's ferret genomic project, hopefully next year the paper will be out, and it will be interesting read. So thank you. You know, it was a lot of reproduction, but I tried to, make it more.
Yeah, well, thank you very much for this torrent of information.
So I know it's a lot, sorry about that.
I appreciate very much this kind of seminars, so we are good. So one question about the first section of the talk. So quite interesting. I ignored this vitrification method to preserve cells. So my question is about the mechanism underlying this. So I'm a plant guy, so my references are from the plant kingdom. So in plants, plants that experience dehydration, of a survival mechanism. So what they do basically is to protect the photosynthetic machinery. That's what they do actually. So that's the most expensive thing to do, plus I guess many other things. But biologically what they do is to protect the precious machinery. So, but you said that the vitrification it's a kind of a glass around the cell, protecting from the environment. But for me, I think it's not enough. So what happens inside the cell?
There are several steps. If you're asking specifically about dehydration, yes. So at least in the species, like where they showed the anhydrobiosis, they showed that, for example, there are several factors that help out. So, for example, trehalose, what it does, it substitutes water in the membrane, because this is the biggest problem, like for cell to be alive. If something happens to the membrane, most likely you lose the cells. So what trachelose does, it kind of substitutes the bonds, it replaces the molecules of the water to make sure that the membranes don't collapse and they still are intact in the mammalian cell. And then inside, at least this is what is described, is that this type of vitrification, which is kind of glassifying stage, is happening, which stops processing, so it's cellular processes. But it's not only the trachelose, there are also, for example, small stress proteins like heat shock proteins and late embryogenesis abundant proteins, intrinsic disorderly proteins, and they work synergistically with trachelose, and some of them are, and there is the reactive oxygen species defense, so they're trying to fight the oxidative stress, they're trying to fight the osmotic stress when it happens. And the biggest problem to replicate it, and what is usually happens in all the species, it takes time. So when they sense that the water content goes down, they start slowly accumulating and synthesizing this trachelose, and so that when the water drops fully, this is when because the trachelose has this glassifying temperature, which at the room temperature, when with enough water lost, it can glassify, so it has this vitrified structure, and it also replaces the water. So it takes time, which is difficult to replicate in the lab. So it seems that it's the time together with the ensemble of all of the factors that protects the cell. I haven't read that they were protecting specific part of the cell, like it in plants, yes, which is very interesting. But I know that the membrane is one of the most important thing to protect, and it does protect it. And then, yeah, you kind of need all of the parts of the cell to check it. But when we are trying to replicate it, these experiments in our cells, we do look then at the DNA damage as well. We do look at the membrane, the intact membrane. We also look at the mitochondrial, for example, health. Are they fine, coming back to life, and is the cell is alive or dead? Like, is it, cutting enzyme?
And which is the success rate when you compare with cryopreservation, for example?
Depends on what you are drying, because in sperm it has been pretty successful, but sperm is not that complicated. It doesn't have that much cytoplasm, right? You can just if you don't need motile sperm, you just need to preserve the genetic material of the sperm, so the head. It's been pretty successful. So, and you could do the IVF, so in vitro fertilization with the sperm, and at least the beginning of the embryo was developing in the cat. So we haven't seen the full development yet, but in the sperm it's working with the oocyte, with the germinal vesicle. So it was, it's not here, but like sperm is successful. Germinal vesicle, this is the kind of the nucleus in the prophase 1 of the meiosis in the oocyte. So this one is been more or less also successful. We managed to do some of the IVF, but again, not full embryo development. And then the tissue is difficult. So right now, tissue is the success compared to cryopreservation is pretty low, because it's a complex tissue. You have different types of cells. You need to kind of preserve the signaling between the cells, the relationship between them, and it's a bit difficult. So cryopreservation still works the best. But yeah.
Yeah, no, I was impressed by the complexity of the network. I guess they really work. I mean, in the sense that when we face a list of genes, I mean, you don't know what to do with them.
Yes. Yeah.
So having these enrichment approaches helps in making sense of this complexity. But the nice things about this network is that you are overlaying several layers of information at the same time. So, but how do you proceed from there? I mean, it's just you go around picking things up?
It takes so. At first, when you start the network, for example, from enrichment result, you don't have these nicely formed clusters that I showed. They don't, but you can infer by the rate of the edge, so the thickness of the edge. So if it's function enrichment, how many genes are shared between it, or if it's the predicted protein-protein interaction, how strong is the so for example, was it also this interaction validated in human or not? So you have kind of the clusters that you can subcluster, and based on this rate of the interaction or strength of the connection between the nodes. And then after that, you really have to go to literature a lot to see if it makes biological sense to cluster them this way. So this is the first step, is to really try to detangle the whole network. And you have some methods that help you. For example, the Markov chain clustering approach helps you to cluster kind of subcluster things, but a lot of this is really the biological interpretation as well. And then you overlay different data. Where do you go from there? It depends what you are researching, right? For example, with the fertility preservation, we found some of the functions that seems to be reacting during the process of preservation. So we then had the functional experiments to follow up with it. For example, we checked the mitochondrial activity actually in the cells. Is it the mitochondria? So we see increase in mitochondrial activity. Is it really increased in the cells, in the follicular cells, or is it just in the stromal cells around it? So you follow up with the function experiment. With the genetic load, I'm not so sure how to follow up from there. I think it's a lot of prediction and trying to see if you see connection between the observed reproductive phenotype. Like, so this is why overlaying this additional information could help us to narrow down which genes or which pathways we should be looking more closer at. And then maybe we can compare it, to in other species, what's happening there in the reproduction, and maybe they're sharing similar mutations.
And I was wondering, from our point of view, we will be generating a list of variants predicted to have some effect, and some of those will be in genes related to reproduction somehow. So our final goal would be to identify critical variants that are causing, reproductive defects, and being able to then manage that based on that evidence. But sorting that out is really difficult.
And what you can do, and one thing that also helps, is starting to generate also expression data, right? You can go to it's easier in male. You can collect, for example, semen, and what we looked at, right, the proteome and transcriptome of the semen. So you have your reproductive phenotype, you have your genetic variants, and then in the same individual, if you have the proteome and transcriptome, for instance, of the seminal fluid, you can see if it's really expressed or not, and if it's actually expressed or not in the ejaculate, so in the reproductive function, in productive cells that you're looking at. It's a bit more difficult with female, how to look at that, but you can, it's the question of kind of having this intermediate expression data that you could then connect to the observed phenotype of the female male, and finally the breeding success. And once you find, with the testing of the genes that you found, it depends if it's species specific, because if it's not, maybe they're already been checked. So for example, in those infertility database, they're all been checked in some other species, and then you also kind of have this. It's not a support, but it's like second, the predicted support from observation, right? That you see it, and maybe this is one of the genes that is you need to look out for, because it was shown as the infertility marker in other species. You see expression change specifically in the reproductive cells of your species, and then it also connects to the breeding success, that maybe this is something that can guide you more towards it, I would think.
A lot of work, anyway.
A lot of work. No, definitely, but also kind of exciting.
A lot of literature searches, yes.
Yeah, it's kind of unraveling all of this and going deeper. It's a lot of fun as well.
So one last question is, how close do you think we are to implement these assisted reproductive technologies in the Iberian lynx? I mean, I understood one challenge was precisely to induce, being able to control the ovulation. Do you think we are close to
I think we are closer than before, because Laura, who is sitting here, starts this year her PhD project specifically on the Iberian lynx reproductive management, and also Jaguar. It's a joint project, the Iberian lynx and the Jaguar, and she already worked on this like the past year at our department, and now she's kind of continuing the work on it. She already did in Silvish tryouts of the artificial insemination. And I think you can, if you would like to say more, Laura, that I think they tried 4 times or 5 times, but this is kind of the This is our chance here to implement it.
In silage, they have tried 6 times with different females, and they were not successful. But we are successful now with the ovulation. So the drugs that we're putting on the females to ovulate are successful. We cannot control when the female is going into estrus, so when it happens, they call me and they say, you need to come in 2 days. So it's still not organized, and that's why Olga's research is also very important, because it's plannable in 2 days. But we do see ovulation, and what we're going to do is we're going to evaluate if not only we see ovulation, but how are the hormones doing. So we're collecting the samples this year to evaluate whether on the female side we're doing okay. On the male side, the males of the Iberian lynx are really not that good. So we are going to try to, the technique that we use is we take the sperm and put it directly next to the oocyte. So we go in the oviduct. So the oviduct is what is between the ovary and the uterus, and we directly take it. So with this technique, hopefully the sperm has to move less, so it will get better. And also we are developing a media that is going to prepare the sperm for fertilization as much as possible. I've been doing this for a year, so I don't know if my opinion is the most valuable, but Katarina, who has been doing this for 20 years, told me, I think we're almost there, a few years, and then we will probably be done. So I'm very hopeful, and now we have an actual team that is actively studying this. My PhD will be about this. I think in the past the problem is that we didn't have someone that was only focused on this, and everyone was doing other things and maybe didn't have time to sit down and really plan the things. So I'm, at least during my PhD, I think it would be possible. For the lynx, for the jaguar, I don't know, but for the lynx, I think yes.
Yeah, and the samples that Laura mentioned are the feces and urine samples that are collected in the breeding centers, and then we can measure them non-invasively, the hormones, and really track the cycle. So those are.
So I understand that the artificial insemination is the closest to become real. But what other techniques do you think could help? I mean,
yeah, we know that there is kind of the new project that hopefully will be starting by Eduardo Roldan from Madrid lab about the oocyte pickup and then doing the in vitro fertilization and doing the embryo transfer. And this can really help in a case where, for example, you have a female that maybe has a problem with the implantation, like maybe some problem with the uterus, and then maybe you can implant this embryo in another female, and this way you can preserve the fertility from both male and females. This is a little bit further away. It's more difficult, this technique. So I think it would be possible. I mean, we'll see how to develop. But again, for example, in cheetah, 5 years ago, the embryo transfer worked, but they tried for a while. It took a while, but it worked in the end. And I know that, for example, in the Thailand lab where I worked before in Bangkok, they also did the embryo transfer in Elk's deer, and it also worked there. So it's a bit more tricky, it's harder to predict it. But it's also gives you more access to more animals, to more genetic material. But that's why we're trying, so there is we'll see how first you try to protect the habitat, then you try in the breeding sense and natural breeding. If that doesn't work, you try to do this artificial reproductive techniques which are more easier. So for example, collecting semen and doing artificial insemination. At first you try, maybe you can inseminate without, maybe you can just inseminate into the uterus directly through the vagina transcervically. If that doesn't work, for example, as in lynxes, then you do the laparoscopy. So you kind of have to do a bit invasive, so you go directly to the ovary, to the oviduct. And if that doesn't work, then the oocyte pickup and the embryo transfer could be the next step and the answer for that. And especially, of course, if you do, for example, some of the cloning, because in the black-footed ferret, that's what they did. They did the cloning and then they transferred it, but to the domestic ferret in their case, and it worked out. So you have to develop this technique for the future. And this is something that we also talk a lot, and together with Laura, that the things developed in the lynx also, and in other, in this case, in the lynx, can be applied into other species. So if you can show that it's possible in the lynx, you can then try to replicate in other species with maybe some adjustments. But it's going to be inspiring the next studies too.
Okay. So
So thank you again for the talk.
Thank you very much, everyone.